Okay, so welcome everyone to today's seminar. Today we have Jared Wilmoth. Is that the right way?
You pronounced it better than anyone ever has on the first try.
Really? Oh.
Yeah. Usually they say Wilmoth, but Wilmoth is like better.
Yeah, Wilmoth. Okay.
Thanks.
We have Jerry Wilmoth. Jerry is a new faculty member, a new assistant professor at UMD in the Department of Environmental Science and Technology. Jared and I have lots of common interests. We both like laser and orbit trap and the geochemical cycle. So let's welcome Jared to give us a talk about his research.
Thank you, Grace. Okay, so I think you might have to enable me to share my screen.
We haven't done it. Can you do that now?
Yeah.
Sure, great.
All right. Okay, all right. All right. So do you generally, during the talks, do you like to see people's, well, I guess everybody else has their videos open, so I'll keep mine open as well. Okay. So first of all, I'd just like to say that it's an honor for me to give this talk today. I just started as a new faculty member with the Environmental Science and Technology Department, and I'm coming from Princeton University, where I was a postdoc for a little over 2 years, working on a methane emissions project from wetlands, and that was with the Princeton Environmental Institute. And I was housed or based out of the Geology department, and so you know it's great to be talking to you all today in the geosciences. And so just a little bit about my background before I get into my talk is, like, I like to think of my biogeochemist, and I'm usually thinking about I usually think about things like framed in redox oscillating systems or soils, for example, that undergo redox transitions. And that could be on large scales or small scales. For today's talk, I'm really going to talk about things like, redox active metals in the environment, or minerals like iron oxides and iron reducing bacteria and organic matter, and how sort of all that stuff in soils kind of interacts. And my focus is on those interfaces and how do we investigate those. And so my talk is really going to be, it's not going to be like a really focused research talk on any one particular project, but it's going to be a collage basically of all the different things I've done in recent years. And really just to give you an impression of what I've done and sort of where I'm headed. And so with that, I'll get right into my talk. So the first project that I want to talk to everyone about today is this project that I had at Princeton, and the title of it I gave this talk or components of it at Goldschmidt in 2018 in Boston. And so some of the topics I'm going to cover up front are going to be from that talk, but then I've thrown in some extra information toward the end that I'll highlight. And so I'm going to discuss this topic called the methane paradox and how it can be linked to redox transitions. So just a little bit of background, right? I think we're all on board with methane being an important greenhouse gas. And if we just think about it in terms of a pie chart and where, where natural wetlands would fall in that contribution of methane to the atmosphere, we can see that it rivals some of the other major sources, like from fossil fuels burning and other things like that. And so it's really important to understand the natural mechanisms of methane being emitted from wetlands. And so, sort of the classic way that we think about methane emissions in wetlands, for example, is, we'll you can think of a saturated soil layer, for example, at depth, and sort of how under those saturated conditions you get long-term anaerobic conditions. And we can think about this, especially so the system that I worked with was a peatland, which is very organic rich. It's a very organic rich soil, much more organic on whole than it is mineral. And so, but it tends to be made up of really complex carbon, like lignin-like compounds that are polyphenolic and really generally hard for microbes to break down under anoxic conditions. But so there's this carbon cascade, I'm going to call it, where in that system, in order to get to methane, you need to initiate the degradation of complex organic carbon like polyphenols, lignin-like compounds, get fermentation byproducts like acetate, hydrogen, CO2, and then C1 compounds like methanol, and then that can feed methanogenic archaea to produce methane. But there's been some really interesting research in recent years that suggests that methane is actually being emitted in several cases from environmental systems from near surface layers or surface layers that are oxygenated. Okay, and so people have found that in certain systems, like for example, the Great Lakes, forest soils in Puerto Rico, that even when you have a soil under oxygenated conditions, those types of conditions can actually promote very high levels of methane production. In some cases, 60 to 80% of the methane emitted from a wetland, like those studied in New Hampshire that I'm calling out here, have been speculated. Those emissions have come from surface layers or near surface layers. And so there's some direct connection between oxygen and methane production, which is counterintuitive, because we generally think that under oxygenating conditions, oxygen would basically kill methanogens or shut methanogenesis down, right? So under the conventional model, we sort of, soil scientists have tended to think that, okay, you need complete saturation in order to get methane emissions from wetlands. But now there's a new emerging model where people are starting to think more realistically about things like anaerobic microsites and oxygenated soil layers as being major contributors, in some cases, the most primary contributors of methane emissions from wetlands. But there's still a lot of questions, and no one has really shown this directly. And more to the point, no one has shown a direct mechanistic relationship between oxygen and methane. So in other words, how oxygen can actually enhance methane production. And so that's what we set out to do with our project at Princeton. And so I was actually working in a group called the Carbon Mitigation Initiative with Princeton to work on this. And so our hypothesis was that redox transitions, or an oxic-anoxic transition, can enhance methanogenesis. And we were really asking this main question: why do significant methane emissions originate from near surface layers, or in other words, layers where there's plenty of oxygen around? And could oxygen play a direct role in stimulating methane production? All right. And so I'm going to talk a little bit about how that's possible. So if we go back to this idea of a carbon cascade, right, like where we start out with really complex polyphenolic compounds, one way that you can break those compounds down more efficiently, okay, in a more thermodynamically favorable way, is by adding oxygen to the system or having oxygen around. Because typically the microbes that can use oxygen to break down carbon, some of them are very specialized at breaking down polyphenolic compounds like lignin and things like that, that you would find in wetlands. And so then that could boost fermentation in subsequent anaerobic cycles, or sort of, if you're in a system that's kind of, facultative between oxic and anoxic conditions, it could feed those processes, creating more methanogenic substrates like acetate, hydrogen, CO2, and C1 compounds. And then that could actually enhance methanogenesis. Okay, so in a redox oscillating system, you could have a situation where you've got oxygen for a while, and then you've got anoxic conditions, and we think that could stimulate methane production. So what we what we did is we went into the field and we picked a field site from Andover, Massachusetts. This was the Ward Reservation in Andover, Massachusetts. Collected wetland samples that came from a sphagnum-derived peatland. Okay, so this is sphagnum moss. There's a picture of it here in the slide. And then we collected peat samples from different layers below the water table, sort of just above, and then further above the water table. And we used serum, like a serum bottle incubation in the lab to do an incubation study. So what we did is we had this experimental scheme. Well, this is the scheme of the experiment, right? So very simply, we had controls in these serum bottles where everything was always anoxic for almost an 8-month period, okay? Then we had another treatment, okay, or we had the set of treatments that involved the different layers of peat that we collected, and they were all exposed to different levels of oxygen, either 5% or 10% oxygen for about half of the incubation period, and then which was 3.5 to 4 months. And then for the rest of the incubation, they were allowed to naturally go anoxic, and then we measured methane production, okay? So these are the methane results. So over a 230-day period, we really didn't see, any methane production occurring until toward the end of the experiment. But once we started to measure methane, the really cool result, right, the remarkable result is that we saw orders of magnitude more methane produced after the oxygen treatments. And so this is oxygen that was flushed into the headspace of the serum bottles to create like 5% or 10% O2 levels in the headspace at the beginning, right, like during this oxic period. And then the controls were always anoxic, and then we've got the treatments following that oxygenation treatment. And so this was really a big result for us. And so the next thing we needed to do was figure out, okay, how is oxygen, directly influencing this result that we see with methane emissions? And so one of the things we did is we looked at the microbial population. So I'm showing sort of this was preliminary data at the time based on 16S ribosomal RNA gene relative abundances, but I also was able to analyze this further with metagenomic analysis to back this up. But one of the cool things that we found was that oxygen it increased the relative abundance in the system of this genus called Novus Vingobium, which there's been a lot of interest in it in recent years, but there's not a lot known about it in wetlands. But the interesting thing about it is it has each genome of Novus Vingobium in the databases has anywhere from tens to hundreds of different genes that are specifically necessary for breaking down certain types of complex carbon, like polyphenols, lignins, and things like that. And it turns out the byproducts of those compounds are methoxylated aromatics. And so we also detected this genus called Holophagia, which its growth was shot up during subsequent anaerobic conditions. And we were never able to prove it because I'll talk about a mass spec technique that we used next to try to look at different substrates in the carbon pool. But we kind of missed it with that analysis. But anyways, there were some implications here that there may have been this really nice metabolic pathway between an aerobe and then a subsequent anaerobe using a specific degradation pathway, ultimately leading towards methanogenesis. And we know that Methanobacterium was likely the prominent methanogen in the system, and that type of genus uses hydrogen and CO2 to make methane. It doesn't start with acetate, which is usually the common pathway. So in order to get at the transformations in the dissolved organic carbon fraction, we use this technique, which some of you may be familiar with, ion cyclotron resonance, or FT-ICR-MS. And so this is a technique that's really powerful, a lot of resolution to be able to see different compounds in a complex sample like a soil extract. And essentially what you're doing is you're injecting the dissolved organic carbon into the spectrometer. It gets trapped, the ions are trapped in the center of a magnetic field. You can pulse radio waves into that ion cluster, and the ions will orbit resonantly inside of the spectrometer. And there's detector plates that can measure the frequency of the orbits. And so heavier things orbit or revolve more slowly, lighter things revolve more quickly, that kind of thing. And so you can get really detailed mass spectra. Okay, and then you can plot based on the information from the MS. You can calculate the molecular formulas just based on the exact mass of each compound detected. And then based on the chemical information, you can plot things like hydrogen versus, the hydrogen to carbon ratio versus the O to C ratio, and get these, what's called a Van Crevelen diagram, where each point in the diagram represents a different compound. And so you can compare areas in the spectrum. So the red and blue dots represent different treatments. And so this is sort of like, you can compare two different samples, all right? And so we had 16 different samples that represented different treatments, the controls, different time points. And what we found, if we just look at a very straightforward principal components analysis of the mass spec data derived from the ion cyclotron resonance mass spec, is everything that clusters on the right-hand side, these are all early time points, treatments and controls alike, okay? And it wasn't until the later time points when we saw a divergence in the composition of molecules detected. Okay, so again, this PCA is developed, or it's derived from relative intensities from the ICR mass spec data, okay? And so at the end of the experiment, we saw large enough differences qualitatively between anoxic controls and treatments that had been treated with prior oxygenation. And it turns out that you can use the weight or the loadings of the molecules, okay, on the principal component axes, and to sort of parse out which molecules were most influencing certain samples at the end of the experiment. And so by doing that, you can make different plots that kind of help you get a sense of what was really different in terms of the DOC composition at the end of the experiment between the anoxic controls and treatments that had been treated with oxygen previously in the experiment. And so one of the things that jumped out was that, by the end of the experiment, under anoxic conditions, like long-term, this is the control, anoxic conditions, that we see clustering in the, these are the Van Crevelen diagrams again. We see more clustering here, and this is a section of the Van Crevelen diagram which indicates microbial proteins and things like that. So this could be the result of cell lysis. This could be lipids from microbial cell membranes and things like that. But then what we see more enriched following oxygenation by the end of the experiment. And again, this is also under anoxic conditions at this point, right? Because it's at the end of the experiment after a redox transition. But what we find is clusters that are more representative of condensed aromatics, polyphenolic and lignin-like compounds, tannins, things like that. And so this is sort of characteristic of the peat itself, okay, at least. And so what it's telling us is that after oxygenation, even under prolonged anaerobic conditions subsequently, there's microbes that apparently are able to break down the actual solid peat to release the compounds that the peat is made up of and sort of give us this characteristic profile. So, it kind of suggests that maybe the oxygen stimulated certain microbes, even at the end of the experiment, to be able to continue to break down solid peat. And so the other thing we could look at is like nominal oxidation state of the compounds versus their exact mass. And so the other thing that we saw is that following the oxygenation treatment, we tend to get smaller molecules, around 400 to 450 Daltons that had a higher nominal oxidation state, whereas with the anoxic controls, we tended to get larger molecules that had a lower nominal oxidation state. And this is sort of in line with the modern view of how organics get broken down in soils, where you start out with really complex things and they continuously get in their, larger, more energy-rich molecules that can be broken down into smaller, more, energy-less yielding molecules. Okay, less thermodynamically favorable, smaller molecules. So there's some other ways that you can plot these things. You can look at the oxygen to carbon versus exact mass. You can look at double bond equivalency per carbon versus exact mass. And just in a nutshell, what we found looking at aromaticity, just to summarize what we found with this data is that oxygenation tends to lead to smaller molecules with higher oxidation state, higher aromaticity, and higher double bond equivalency per carbon. And I guess the one thing that I wanted to mention just before I leave this completely is that any of you that work with this type of mass spec data, I'd really be interested to get your feedback at the end of the talk, because this is, the right way to go about analyzing these things, and sort of the way you interpret it, I think it's still something that's not, it's not very well it's just not very well tackled, I think, in recent literature. So if you've got any insight on that, that would be great. So the other thing that we looked at too was the gases produced in the headspace of the serum bottles, like CO2 and hydrogen, which again, based on our metagenomic information, keyed us into the fact that the methanogen predominant in the system should require CO2 and hydrogen directly from the headspace to make methane. And what we found is that oxygenation, the oxygen treatments, always led to higher levels of CO2, okay, across time, versus the anaerobic controls. So right there, that's a key substrate for methane production that oxygen promoted directly, all right, which would also be linked to how things are being degraded and metabolized in the DOC pool, which I just talked about. And then hydrogen is also an important substrate, right? So for every mole of CO2 that's consumed to make methane, you need 4 moles of hydrogen gas. And so we always saw like relatively more hydrogen in the anoxic controls, but we ended up seeing more or less hydrogen, we think, in the treatments because of the requirement because you need 4 moles of hydrogen to make methane using CO2. So for every mole of CO2. So, just using this type of like net analysis of these gases, we're kind of losing some insight on, the flux or the rate of how these gases, like how fast these gases are produced and consumed, because there's different reactions that could consume these gases as well as produce them under anaerobic conditions. And I think using isotopes of carbon especially would be a really great next step to sort of include in this type of work. And so the conclusion was that, our results indicate that oxygen enhances methane production during anoxic conditions. And we used a temporal approach because other people have sort of like put this in the framework of wet dry cycles in the environment, which is great. But if you want to do this in a laboratory setting and try to, cut down on as many variables as possible, our system was really great because we just had a slurry and a serum bottle with the peat, where there wasn't any wet and dry cycle. We were just adding oxygen. And so we were able to make a connection just between oxygen, not without wet and dry cycles, to show that oxygenation during redox transitions can cause more methane emissions. And so, of course, that has implications for the field, how wetlands emit methane, the rates at which they emit methane, depending on redox transitions. And so I'd just like to acknowledge the people that I worked with really quickly at Princeton. And so one of the things that I've been talking about today is sort of like the biological pathways of methane production. But we were also thinking about abiotic mechanisms, right? And so one of the things that we started to think about and we started to pursue, but didn't get very far, was thinking about, okay, if there's iron in the system, we could potentially have abiotic mechanisms starting with Fenton reactions, okay? So if we've got iron 3 and iron 2 and oxygen in this system where there's a lot of organic matter, Fenton reactions are essentially this abiotic reaction that creates different oxygen radicals. And those radicals could potentially break down the carbon in the peat, just like the microbes, and essentially fuel methane production. And so we wanted to try to just investigate the iron in the system. And so what we did is I went to Brookhaven National Lab with some collaborators, and we used a synchrotron at Brookhaven, the National Synchrotron Light Source version 2. And we were able to use X-ray absorbance near edge spectroscopy to sort of map out these hotspots of iron. And I was able to use Mossbauer spectroscopy, although I'm not going to show that here, to validate that these are probably nano iron oxides in a mixed valent state. Okay, so you got iron 3 and there's iron 2 mixed in there as well. And so this spot size, so all of this blue area is the peat matrix. Okay, it's the organic stuff. This is the iron hotspot, and this area is probably 20 microns. Okay, and when I say that this is a nano iron oxide, that's based on the Mossbauer evidence. And even though there's just one blob here, I'm not thinking that this is just like one isolated iron oxide. This is probably some aggregate of smaller iron oxides. And so anyways, we were able to show at least that during oxygenation, which is where this data was collected, it was a sample that was undergoing the oxygen treatment, that we did have mixed valent iron, and the conditions were right to have Fenton reactions. But we never got far enough along to be able to pursue the Fenton reaction theory further. And so this is something that, you know, is sort of like, I'm always thinking about, how does iron factor into these kind of things? And so could be a great pathway for future research, especially in wetlands. And so now what I want to do is use that iron presentation, or those slides on iron, to sort of segue into what I did for my PhD research. So now I'm going to start talking about my PhD research at the University of Georgia. I finished up my PhD in 2016, and I was working with these soils in Puerto Rico. Some of you may be familiar with the critical zone observatories across the United States. This one, they call it the LCZO, or the Luquillo Critical Zone Observatory, in the Luquillo Experimental Forest in northeastern Puerto Rico. And so, you know, there's a lot of carbon that these soils store, and you know, these soils in Puerto Rico and the rainforest, they emit some of the, you know, or have some of the highest carbon turnover rates that we know of. And one of the other cool things is that these soils have been studied for years, possibly decades, just because of the interest in how quickly they go through redox transitions. So because of the periodic rainfall, you can get very rapid redox oscillations, even in the mountains. And so anyways, we were working in the Beasley watershed in the Luquillo Experimental Forest. And so this is some oxygen sensor data from the field site that we worked at. And this is from, so each line is just different sensors collecting data on the oxygen levels and what we, over time. And, you know, so what you can see is that, number one, there's a lot of heterogeneity in oxygen levels. And if I'm right about this, I think all of these sensors were probably at the same depth, just in different physical locations, like close together. And so what you get an impression of is just how heterogeneous oxygen levels can be, you know, and that there are these natural redox oscillations, okay, from, you know, low oxygen to higher oxygen to lower oxygen again. And that even within those larger oscillations, there are these smaller undulations or oscillations in the oxygen, which also means in the redox potential, okay? And that's going to impact the biogeochemistry and ultimately the broader, carbon emissions and things like that of the system. So we were looking at this from a more fundamental level, right? A more iron-centric perspective, because iron is like this biogeochemical hub, right? It interfaces with organic matter, with bacteria, it influences carbon. And so, one of the well-known sort of chemical aspects about iron oxides is that if you just let them set, if you take an amorphous iron oxide and let it set for a while, over time, it'll tend to reorganize itself into a more thermodynamically stable crystalline form. Okay, another term for that is Ostwald ripening. So what we know, or one of the things that we know very well, is that if we have a solution of iron to ferrous iron and we oxidize it very rapidly, okay, and this is in a clean system in the lab, that slow or rapid oxidation will generate iron oxides that are, amorphous, okay, generally. But if we oxidize very slowly, we can end up getting more crystalline forms of iron. But now, how this actually plays out in a complex sample like soil, nobody had really investigated that very well in this particular way, like in the framework of redox oscillations over time. And so what we wanted to do is we wanted to characterize that type of mechanism, all right, because it has big implications for the carbon cycle, especially from these Puerto Rican soils. So in order to study iron valence in iron chemical environments, a great technique is Mossbauer spectroscopy. It's, arguably one of the most sensitive analytical techniques known. And because it uses really high energy radiation to get information, all right? So basically the setup that we had in our lab was a cryostat, which was just a cylinder that we could control the temperature anywhere from room temperature down to 4 degrees Kelvin. And we do that by compressing liquid helium. And this is a sample rod that would just hold a soil sample, all right? And then on one side of it, you've got a source. It's cobalt-57, which is radioactive. It emits gamma rays with this energy. And by oscillating it back and forth, you can get a Doppler shift in the energies, just enough to sort of probe the different energy levels of iron-57 in a sample. So it's not iron-56, it's iron-57 that has these the right nuclear spin characteristics to be able to utilize this technique, because the energy levels in iron-57, the iron-57 nucleus can change depending on resonant absorption of the gamma rays. And so what you get when you do that is you can get basically, whenever they plot Mossbauer spectra, you've got gamma counts, or like the intensities, right? And so you've always got a baseline, or I'll go ahead and explain the x-axis too, which is also sometimes confusing. It's plotted in velocity, because this is talking about the velocity relative to your sample, or relative to the detector, of that sort of that oscillating motion of the source, right, to emit the gamma rays. But we can think about this as energy, okay, where from left to right, we've got low energy to high energy energy transitions between the iron nuclear states, like the ground state and the excited states. And this is just the relative intensity. The reason why the spectra looks upside down is because this is the baseline gamma counts going to the detector. And every time the sample absorbs gamma rays, you're getting a dip or an absorption dip to create the spectrum, all right? Now, if we're thinking about our, what I call the biogeochemical hub, okay, with iron at the center, if we've got an iron oxide and we've got microbes, and there could be iron too here, certain peaks in the spectrum are representative of iron 2, paramagnetic iron 2, paramagnetic iron 3, which would be these, what we call doublets. But then there's another set of 6 peaks called a sextet, which represent crystalline iron oxides. And the cool thing about this is this spectrum was recorded at 140 degrees Kelvin, so relatively cold compared to room temperature. But so the interesting thing is that you can take a paramagnetic iron crystal, and if you cool it down, you can relax those, you can relax the thermodynamic instability in the mineral, or, you know, because of the thermal vibrations. You cool it down and you can actually induce a magnetic field over the mineral, and that magnetic field will lead to these features. So what that means is that really crystalline iron minerals will show this sextet type of signal at room temperature. But paramagnetic iron minerals that would give a signal like this doublet, if we cool it down enough, like down to 4K, for example, this signal would also convert into a sextet. And that tells us something about the degree of crystallinity, the different pools of iron. And so this is a really powerful technique. And so what we did is an experiment. What we did is we collected soil from the Puerto Rico field site, okay, from the rainforest in Puerto Rico. We took soil from the site from one layer profile, and then we put it in serum bottles, just like I discussed before for the wetland methane project. And we created these soil slurries, and what we did in order to, this was all in the laboratory, in order to impose redox oscillations, you know, not exactly the way that we've seen in the field, but like something like what we've seen in the field and something that we could manipulate in the lab, is we would allow the system to go anaerobic. And so we're looking at iron 2 concentrations. So remember, whenever we see the lines go up, the data points go up, that means iron 2 is increasing in solution or in the solid phase of the slurry, which means that anaerobic conditions are underway, iron is being reduced. Okay, iron 3 is being reduced to iron 2. And so we have an anaerobic period followed by a small oxic period. So it's like 7 days of anaerobic conditions and then 1 day of oxic conditions, and then that cycle repeats. And we can see that by doing that, we were able to induce these redox oscillations in the iron pool, in the aqueous. We can measure that by the aqueous phase iron in the solid phase. So the solid phase is just taking the soil and extracting it with an acid to pull out the acid extractable iron 2. But what we did for treatments, and it's subtle, but it's important, is we use different oxidation rates. Because thinking back to that initial slide where I showed the sort of a cartoon of redox oscillations with iron, and that slow oxidation can lead to more crystalline iron oxyhydroxides, and faster oxidation of iron 2 can lead to more amorphous or poorly crystalline or poorly ordered iron oxides. So that's what we were trying to test, right? And so the slow oxygen treatment is that during every oxygen step, we were injecting 1 milliliter of oxygen every hour over a 7-hour period. So that was just 21% oxygen to create like, so that was like, I forget exactly how the numbers work out, but we did it in such a way that the oxidation rate should have been about an order of magnitude difference between all of these, okay? So we've got a factor of 1, a factor of 10 for the medium, and then a factor of 100 for the fastest oxidation. So what we found is that if we look at the Mossbauer data, and we sort of calculated, or we're presenting the Mossbauer data so that we can just look at the iron of highest crystallinity, so the amount of crystalline iron as a percentage of the total iron in the system. All right, so these bars just indicate the amount of what we're calling crystalline, or the most ordered iron in the system, right? Iron oxyhydroxides. And I should say that the predominant iron oxyhydroxide, as far as we could tell, was what we would call nano-goethite, okay? And so if we look at the iron in the initial stage of the soil, and this is before any treatments were imposed at the very beginning, this is about how much crystalline material was there in terms of the iron oxyhydroxides. Following the fast oxygenation treatments, we saw an increase in crystallinity, okay? And we saw the most increase in crystallinity following the slow. So this is in line with what we would hypothesize, right? That even in a complex soil, what we're seeing is that, or a complex soil sample or a slurry, that when you oxidize iron after repeated cycles, okay, very slowly, you get more crystalline iron building up. But we also saw the same, we also did see a little bit of an increase even after our rapid oxygenation treatment, which means that there are soil processes, or, you know, maybe our oxygenation treatments weren't powerful enough to really sort of drive it in the other direction, where we would see more crystalline material building up. But this is kind of interesting, and I don't have a great answer for that right now. The other thing that we were able to do is, because Mossbauer is sensitive to the iron 57 isotope only, you can spike a sample with iron 57, and you can actually, this is something that I've never, people handle isotope enrichments differently in the way that they apply Mossbauer spectroscopy. I tried to do something that I felt was different than anything I've seen in the literature, where I was actually, in each Mossbauer spectrum, the black dots, everything under that represents 100% of all of the iron 57 in the sample. Basically 100% of the iron pool, if you want to think about it that way. And everything that's shaded, everything that's collared here, is the 57 contribution by relative abundance from the iron 57 that I added to the system. And so if I go back just really quickly, so what we did is we did another experiment offline where we added iron 57 sort of at the peak point in iron reduction, and then we oxidized it. And so the data that I'm going to show you now with the isotope information was collected just after adding the isotope as iron 2, iron 57, and then oxidizing it. I'm going to show you that data, and then I'm going to show you what happens after it's been reduced for 7 days, okay? So after we've added aqueous iron 2, 57 to the system, this is what it looks like when it's been oxidized. So most of the material, most of the iron 2 has been oxidized into a paramagnetic iron state. So really amorphous. This could be iron 2, iron 3 that's just, I should mention too really quickly, everything that's orange is iron 3, everything that's green is iron 2, all right? So what we can see is that most of what's been oxidized has been oxidized as poorly crystalline paramagnetic iron material, with a little bit of crystalline iron showing up in this sextet area. Now, after reduction, after a reducing transition that occurred for 7 days after that, what we see is that most of this poorly crystalline iron has been converted to iron 2 through microbial reduction, which, you know, that's pretty consistent with what you would expect, right? The interesting thing is that during the reduction of iron 3 to iron 2, we also see an increase in the iron 3 oxyhydroxides under the sextet, which means that during reduction of iron 3 to iron 2, we're getting an increase in the crystalline iron, the crystalline iron 3 oxyhydroxides. And I have some, I can speculate on why that is, and you can ask me about that further if you like. I was also able to, at the end of the experiment, collect samples and do a meta-transcriptomic analysis for the microbial populations. I was looking at a lot of different things, but I wanted to home in especially on particular genera that we know are important for iron reduction. One of the most well-known, arguably the most well-known, is Geobacter. And then another one that's less well-known is called Pelicynus. And I can come back to this information, but I just wanted to at least point out that I have a meta-transcriptomic profile of the system to link to the geochemical data. And there was some cool results, but I just don't have enough time to talk about it today. And so I want to acknowledge a lot of people that I worked with, and in the interest of time. So I'm almost finished with my talk. Looks like we've got about 15 minutes left before 1 o'clock, but I've just got a few more things I want to talk about. Project that I worked on at Oak Ridge National Lab. So I finished my Ph.D. in 2016 at the University of Georgia, looking at these soils from Puerto Rico and iron redox dynamics. And then the obvious thing is, if you're a soil scientist, you can't see anything. Everything's deduced based on chemical information or biosignatures or whatever it is through omics. But I wanted to try to figure out ways that I could see things on a smaller scale, right? Actually see things and physically physically probe like spatial characteristics of how we think soil microsites might exist. And so right here is this is a picture of a little, so I'm going to call this a lab on a chip. And I think there's actually a journal called Lab on a Chip, where people that are into microfluidic design and things like that, they will publish in that kind of journal. And this is an example of a very simple sort of microfluidic system that we developed at Oak Ridge, where you could seed microbial cells into little micro wells, okay? And this is on a microscope stage. And so we designed these lab on a chip sort of platforms, okay, using a microscope glass cover slip. And we could use different types of epoxy resins and photolithography and things like that to develop these different micro well arrays. And you know, the cool thing about this is you can literally just about any kind of experimental design with replicates and different blocks, treatment groups that you could imagine, you can implement this all in a on a microscopic scale, literally on a microscope cover glass. And then, so what we could do is we could seed different populations of bacteria. In this case, and this work was published in Frontiers in Microbiology, where I'm the first author. And here it's indicated as 2017, but it's actually 2018 in Frontiers in Microbiology. But we had 2 mutants of Pseudomonas aeruginosa, which is like people study it a lot because, you know, it's found in the medical field as a contaminant, but it's also forms really robust biofilms in the environment and things. So it's kind of cool to study from the perspective of biofilms and environmental interfaces. So the green cells that we had growing in these little micro wells, which were, you know, I think, well, you can get a sense of the scale, like how big they were by this cross section up here, or this diagram. But the green cells were essentially, were Pseudomonas aeruginosa that were just wild type, just the natural form of the bacteria. And then we had a mutant version, which is indicated by the red cells, that we called the killer cells because they were mutated in a way that allowed them to kill the other cell, okay? And there was a mutation in their type 6 secretion machinery that allowed them to kill the other cell. And so we just wanted to see how, you know, how do things play out when one organism can compete with another one through cell lysis and things like that. And so this is just one thing that we worked on. We also used agent-based modeling or individual-based modeling simulations. I used to have a little button here that I could click to run a cartoon of one of our simulations, but it tends not to work a lot, so I didn't put in the cartoon. And this is the last slide that I'm going to talk about. And so going back to the things that Grace is working on and Ricardo, one of the things that we were edging towards, and I really just got started to work on this at Oak Ridge before I transitioned to Princeton, was, okay, if we take these little lab on a chip platforms that we developed, we could actually make the underlying substrate, instead of a glass slide, we could have a layer of, well, you can still have a glass slide, but you can incorporate cover slips. That it's a cover slip, not a slide. You can order glass cover slips that are coated with metals. So like we had ones that were coated with Indian tin oxide, so ITO coated slips. And so then it's electrically conductive. You can look at it under an electron microscope and things like that. But anyways, this is an electron micrograph of a geobacter cell at the bottom of one of our little micro wells. And it's mixed with a population of Dihalococoides McCartyi cells, which are really, really tiny. And you can see some of them actually are, you know, sort of absorbed or stuck to, or maybe interacting with the geobacter cell. And so one of the cool things about this relationship is geobacter produces coronoids like vitamin B12, and vitamin B12 is a cofactor that's necessary for Dihalococoides to be able to reduce toxic halogens in the environment. So it's a cofactor, B12 is a cofactor for the reductive dehalogenase of Dihalococoides McCartyi. And so what we were able to do is use MALDI-TOF-MS, or matrix-assisted laser desorption ionization time of flight mass spec, to be able to scan over these microwell areas, to raster over it with a laser, and then, try to pick up the signals from vitamin B12. Again, this work was really cool. We were just edging into it, and it was sort of what we were doing next to follow up on what we had done previously with the microwell arrays and modeling. But that's kind of where it stopped. And so these are things, I'm going to finish my talk there, but I just want to say that all of the things that I've talked about, I hope that you can see that it's following this research arc or trajectory where I'm really trying to conceptualize the complexities of soil at a small scale. And, how can we use like these bulkier techniques to deduce what's happening at mineral microbe interfaces? But then how can we actually use techniques, for example, like laser ablation, secondary ionization mass spectrometry, these sorts of things, or X-ray absorbance techniques to actually try to map, like develop chemical maps and things like that at small scales. So, and then ultimately to see how everything connects to the larger picture, right? To the ecosystem scale, to greenhouse gas emissions. So I'll leave it at that. I have went over a bit, maybe just a bit, but if you guys have any questions for me, please.
Thank you for the presentation. There are a lot of overlap between your research and our departmental interest. And then I will just let people ask questions.
Sure.
Go ahead.
I have a question.
Sure.
Yeah.
I raised my hand, but apparently she didn't. I have actually several questions to ask you, like 2-3 questions from the first part and then 2 questions from the second part.
Sure.
For the first part, what was the temperature and pH and salinity of your incubation? When did you do first your the experiment that you did with oxygen, like the anaerobic, then aerobic? What was the temperature, salinity, and pH? I think especially salinity and pH could have very good impact on your result, like your observed result.
Yeah, so do you mean specifically with the wetland project that I discussed, or
the one that you did the incubation? Yeah.
Yeah. Okay. So this was the one. I also had the one from my PhD with the iron, and I'm not sure if that's
the first. I'm talking about the first one.
Okay. All right. Yeah. So the pH, peat systems or peat derived wetlands typically have a low pH. It's actually very low, like compared to other environments. So the pH in the of the incubation was 4 on average.
Okay.
But yeah, it was on average, it was about 4.0.
Okay. And how about the salinity and temperature when you did your incubation? And did you monitor it? Like, did you monitor during the time that you were incubating, like during, for example, 200?
Yeah. So the pH was monitored.
Okay.
And so, like I said, on average, it was about 4.0, but from the field, it was lower than that. It was it was anywhere from 3.7 to 3.9. Okay. And then toward the end, it got up to like 4.2, but on average, about 4.0. For the salinity, we did not track the salinity, but I will say that this wasn't a wetland that was impacted by, it's not a tidal wetland. So it's not a marsh where there would be an influx from salt water, basically. But I don't know what the exact salinity is. And then your other question had to do with, what was the other?
What was the temperature?
Temperature was just standard laboratory temperature. So it was, I'm guessing it was, 22 degrees Celsius in the room.
So you kept your experiment, your incubation for like all 200 days. It was like 22 degrees centigrade.
Yes, it was in an incubator. Yeah,
it was an incubator. Yeah. So the question that I had about the salinity, because obviously, like any reduction, oxidation element, like anything that could change the salinity could also have effect on the, like I think could have effect on your like experiment. But yeah. And my other question is that, do you think like if instead of oxygen, you like if you have like anaerobic condition and then instead of adding oxygen, you add, for example, nitrogen, could have the same effect because it
something like nitrate is a
methylamine, so it can produce methylamine and then like makes more methane.
Yeah, no, I think that's an awesome question because there's different things that can be oxidants or reductants, and so the group that I was working in, I was actually kind of the oddball because I was working on methane and sort of like this redox oscillation framework, but everybody else in the group works on biological nitrogen fixation mechanisms and things like that. And so I know that right now, my old advisor at Princeton is actively working on the different organisms that have, function in the nitrogen cycle. And so that's where the research is going. I think it's a great idea. And it seems like that's where my old group is headed right now. So I guess keep your eyes open. Maybe they'll publish something.
Yeah. And can I have two other questions from the second part of your talk? If no one else. Sure. Okay. For the second part, in the natural soils, in the soil environment, what is the origin of the oxygen radicals? What produce oxygen radicals?
Yeah, so like I said, I said it's an abiotic reaction, right? Which, I mean, by the textbook definition of the Fenton reaction, it is, but you need to have iron too in order for it to work, right? Yeah. So in the soil, you would have microorganisms generating iron too under anaerobic conditions. And then that iron too can then react with oxygen, molecular oxygen from the atmosphere, and then go through the rest of the cycles or the chain of chemical reactions to produce the radicals. And then the radicals can directly attack carbon, organic carbon molecules.
Okay, and another question for the iron experiment that you did you also calculate the isotope fractionation of iron, like besides like in your systems? Yeah, the one that you did.
Yeah. So you mean like the
enrichment, for example, of iron 57 to like in your systems, like the fractionation when like in your systems between Fe3, Fe2, yeah. So
there was a visiting scientist in my lab at the time named Viktor Tishchenko, and he's published, our group has published work on isotope fractionation. Okay. Michelle Scherer at Iowa State has also collaborated with us and she's done a lot of that work too, her group. But I did not calculate isotope fractionation for my experiments. And part of the reason is that I know that based on like the enrichment of iron that I did relative to natural abundance iron isotopes, that I'm thinking that wouldn't have been a significant factor in the mechanism that I was looking at.
Okay, and I have one more last question from the first part, if it is okay. Can I ask? Yeah. So for the first part of your incubation experiment, do you think adding oxygen, like having the anaerobic incubation and then adding oxygen could also have an effect on the anaerobic methanotrophs? So that could be also the reason that you are seeing it's not just the methanogens that are producing methane, but also not methanotrophs oxidizing methane. So that you don't know, is it the production or is it the less oxidation?
No, that is an amazing question, because what this does is it's taking my simple system that's in a bottle, allowing things to build up like carbon dioxide and CO and methane. But now when you put that into a real system where, like you're saying, you can have methane produced, for example, in a microsite or a saturated soil layer, but then it has to bypass this oxygenated zone where you've got methanotrophs who could convert the methane into CO2 or something like that? So that's a great question. And so one answer, at least in terms of what we found and its implications, is that I think our work still implies that even if you put our work in the framework of a real system, that what it means is that oxygen is going to stimulate methane production, okay, through the mechanisms that I discussed. And I do think that it will lead to higher methane emissions, because I also pointed out, the whole thing that sort of sparked our interest, right, were these more recent observations of high methane rates in oxygenated soil layers. So I think the connection is real, all right, and the mechanisms we provide are real and can help with the modeling of this in the field. But I also think, like you point out, that even if you're getting really high methane yields, we saw orders of magnitude higher versus the control, but maybe in the field, maybe it would just be a fraction higher, because of if you've got active methanotrophy, you could be converting methane to CO2, but CO2 is also an important greenhouse gas. So maybe it all sort of,
But in general, I really enjoy your talk. Great talk. And I will be in touch with you, because I'm also working on methane coming from wetlands. So I will be in touch with you.
Awesome. Yeah. Thank you so much.
Thank you.
Thank you, Jerry, for the presentation and the talk. I actually also get chat message from one of the faculty member. He would like to talk to you more about research ideas.
Sure, that's great.
Yeah, thank you. Thank you everyone for joining today's seminar, and hopefully see you next week.
Thanks a lot, everyone.
Thank you.
Thanks.